All right, hello everyone. Welcome to our very first and hopefully not last episode of Kitchen Science. My name is Rita Blake. I am the Director of Education for the California Nanosystems Institute at UCLA. And so today what we're going to be doing is we're going to have 3 different sets of demos, all revolving around the topic of polymers. So these are going to be experiments that you guys will afterwards be able to do at home, and we'll be recording this video and also posting it online so that if you want to do it yourself at home and follow along later, you'll be able to. So with all of that being said, I'm going to turn it over to our first presenter, which is Liv.
Quick introduction. Hello, everyone. My name is Liv. I'm a graduate student at the UCLA Department of Chemistry and Biochemistry, and I've been working with the California Nanosystems Institute Nano Outreach for about 3 years now. This is our 4th. So I'm really happy to be able to have this opportunity, a little bit different from what we normally do in classroom and summer program outreach. But yeah, hopefully you guys can kind of see my screen now, and we're going to begin with just a brief overview of what are polymers and biopolymers, and then we'll get into the demos and experiments, and then we'll finish by discussing a little bit of current research and applications. Some of you already submitted questions asking, what do you do with biopolymers? What does it do in society, and what is current research like? So we'll try to answer those questions as best as we can. So I'll just jump right into it. What you can see on the screen here, it says, what do these objects have in common? And there's a variety of objects, things that you might think of as maybe rubber, like tires. We have plastic bags. The frying pan here, we're really specifically highlighting the coating on it, or Teflon, to make something non-stick. We have something that's like a hard plastic, as well as a fabric, like polyester. And as well as styrofoam and floss, which is kind of similar to that non-stick feel of Teflon. So as you can probably guess, the thing that these have in common is that they are all polymers. So most things that you typically think of as a plastic is in fact made of polymer, and things like polyester, it's kind of in the name there, giving it away that it's a polymer. And one of the reasons that we have so many different, so many different materials that we make out of polymers is that you can process them to have a variety of different properties, whether you have something kind of like a gel or a paint, or something like a hard plastic that keeps its form. These are different ways of processing polymers. So what a polymer is by definition is anything that's made out of repeating chains of monomers. So in the name, once again, mono meaning one, polymer meaning many. You have these individual units, and then through some sort of reaction, generally either a chemical linker or perhaps a process because of light activating the chemicals, you string them together in these long chains, and they can form molecules that are quite large. So the chemical picture of this, once again, is that you have a monomer unit, and then through a chemical reaction, you link these into a repeating chain, and this can be hundreds of thousands, maybe even millions of units long to form these long molecules. So to kind of look at the sense of scale of what a monomer is in respect to the objects that we use, for instance, the way to think about it of just how small nanoscale is to take the example of a house, for instance. If you estimate that this is about 10 meters in width, you think about how small an individual strand of hair is across, that's about 100,000 times smaller. A nanoscale object is then another 100,000 times smaller. So it's like the difference between that hair and a house would be something like a carbon nanotube diameter and that hair. So monomers themselves can be very, very small, and we link them in these repeating chains, like I said, to make the larger macromolecules of polymers. So not just in commercial products that we use like tires and plastic bags, but there's another key place that we interact with polymers on a daily basis, and that's in biology. So the sort of framing question to think about what biopolymers then are to think, what would your life be like without polymers? So something that a lot of people say, just as sort of a trivia or a fact, is that your body is actually made of mostly water. And this doesn't just apply to things like blood or liquids that we usually associate with something that's a fluid that's mostly water. This also applies to your organs. So a brain or your kidneys that you think of as being solid are very predominantly made up of water. And so the question then that we pose is, how are these things that are mostly water, even something like 83%, a solid structure as opposed to being a fluid like water? And the answer has to do with biopolymers. So biopolymer, pretty much the distinction here is that as opposed to something synthetic that we make into products like plastic bags, it's produced by a living organism. And so these can have a range of properties, whether it's something squishy like gelatin. You look at your ear and you notice there's a difference between the collagen part that's more soft and flexible, and then the cartilage, which has this really firm structure, as well as things like chitin, keratin, which is involved in giving your nails and your hair structure, as well as in animals that have antlers. It's the same biopolymer there. So the key thing here is that polymers provide structure for these things. But you also find polymers in places that maybe you weren't expecting. So for energy storage, starches, what it comes down to is that you're linking these repeating units of sugars, and that creates the starch, which then can store energy, not just for animals, but plants as well. And then as well as something like DNA and RNA, where you're storing genetic information. If you look at them, you see this repeating motif where you have these DNA base pairs. So technically, this is also a kind of biopolymer. So we've talked about different types of materials that you can make the biopolymers out of, but now I'm going to look at how if you have the same material, you can have a variety of different properties just by the environment it's around. So the example to frame this is if you have something like paint. Chemically, the difference between wet paint and dry paint is not, there isn't a difference. You still have the same polymer molecules, but the difference here is that in the solution, you have other molecules that allow these polymers to flow freely, and then when it dries, if you've ever like had a painting palette and you have to poke those dry splashes of paint, you notice it's kind of gummy. This is an instance where now the polymer is really dense, and so it has a more of a solid structure, but chemically it's the same. Another form that polymers can be used in, besides just changing the density of them, is whether or not they have other molecules in the environment interacting with them. So in this case, a lot of our examples that we're going to go through are actually hydrogels. And what this means is that you have a polymer, but then the difference of whether water is incorporated into their structure or not completely changes the physical shape of it. And it can go from something like a polymer, or a powder to a gel. So maybe you guys have played with some of these dinosaurs. I don't know, I had them as a kid. You dump them in water and they expand a lot greater than their original size. Hydrogels can absorb a lot of water, up to 40, maybe even 400% by mass. And then even though they're up to like 90% water, they still have this physical form that is a solid. And we're going to talk a little bit about the chemistry of how something like this is possible with an example that you guys have probably used at some point in your life. And Diane is going to be showing you a little bit more about how that works. So take it away, Diane.
All right, everyone can see me okay? Okay, hi everyone. My name is Diane and I'm a third-year chemistry PhD candidate at UCLA. I do a lot of work with organic materials and making materials for electronic applications. But Liv just actually told us about some interesting properties with hydrogels and polymers. So we're actually going to be taking a look at some polymers that we deal with that all of us probably have dealt with at one point in our lives. And that is actually looking at a diaper, okay? So I know it's a little silly, and this is a clean diaper, just to let everyone know. But one interesting property that diapers have is their ability to absorb a lot of moisture. So I'm actually going to pour some water directly into this diaper. Let's just see how much it absorbs. Okay, so you can see I just poured some water directly on there. And if I flip it upside down, you'll realize that there's actually no moisture that comes back out of it. So I already had this diaper cut open just to, for viewing purposes, but let's actually see what happened inside of the diaper. So you guys will notice that there's this mushy stuff that kind of looks like snow, okay? So this is actually a polymer material, and it's called sodium polyacrylate. It's known to be a super absorbent because it's capable of swelling up to 400 times its volume when it interacts with water. Let's look at a diaper beforehand to see what it actually looked like before we did that. So I'm just going to use a colored piece of paper to kind of give you guys a better view of what comes out. So I'll do the same thing. I'll flip it upside down and just kind of shake it out. Once again, that was a clean diaper, and just cut that open. And you'll notice that there's all this white powder that came out of there. So this is what our sodium polyacrylate actually looked like before we added any water to it. Now, let's take a look at some sodium polyacrylate that is lab grade. So this is, you guys can see it there, sodium polyacrylate. It says it on the bottle. We ordered this from Amazon, actually, so you guys can actually get similar materials from there. And we're going to experiment with it absorbing water. So I'm going to just add about, a tablespoon of the sodium polyacrylate to this jar. So that's about half, and then another half. Okay. And now we're going to add our water to it to see what happens. So if you guys just take a quick look at what the material looks like, it's very powdery. Okay. And now I'll add some water to it. And just give it a little bit of a stir. And just wait a couple of seconds here, and we'll see what happens to it. So I'm just going to stir it a little. Okay, so you guys are actually seeing that a gel is forming. It might be a little hard to see it there, but if I turn it sideways, you guys actually see that I've formed a solid gel from this sodium polyacrylate, and now it can't come back out. So that's the same thing that it was doing in the diaper. It turned into a gel and was capable of absorbing up to 400 times its weight in water. So I printed out some diagrams to kind of show you guys how that works. I think it was a little easier for me than the PowerPoint. But we can imagine the sodium polyacrylate as a negatively charged polymer backbone that's balanced by positively charged sodium ions. So if we thought about a cartoon of it, it would look like this, but the molecular structure actually has this blue sodium polyacrylate backbone and then these positively charged sodium ions here. Once we added the water to it, however, we actually displaced some of those sodium ions. So as you guys notice now in this diagram, the water molecules have moved in and the sodium ions have moved out. So the polymer was capable of becoming a hydrogel and swelling with all of that water. Okay, so we figured out that that happened when we added our water to our sodium polyacrylate. But now I have a question for you guys. We started with sodium polyacrylate, we added H2O. What do you guys think would happen if I added salt to this? So NaCl, sodium chloride. What do you guys think will happen if I just throw some salt in this mixture? What should it do to this gel? Okay, so we're seeing a couple of different responses here. Okay, remember that I am going to be adding salt. We had sodium ions in there that got displaced because the water favored interacting with the backbone of the polymer instead of the sodium ions. So let's see what's happening as we add some more salt into this. So I just poured some salt in there. Okay. And I'm actually, for this part, I'm just going to cap it and give it a little shake in there. So a lot of people are saying that it will liquefy, which is actually our answer here. So might just need a little bit more salt for this. So this is about 3 tablespoons of salt that I'm adding back to it. And it does just take a little bit of time. But I'm shaking it around now. So if you guys look at that, you'll notice that it's actually turned back into a liquid. And just for comparison, I have another one that I made earlier today. So this is the gel that solidified, right? I can turn that upside down just like that. And then this one actually turned into a liquid. So the reason that that happened is because I introduced an excess amount of sodium chloride ions. So if we think about a third picture of what's occurring, the sodium chloride ions are coming in and water is actually favor interacting with them. So the water comes back out of the hydrogel and favors dissolving and interacting with the sodium chloride. That's what causes it to liquefy and actually come out of the polyacrylate backbone. So, and one last example that I can show you guys, I don't know if you guys have ever dealt with this before, but we have these Orbeez here, or water beads. And sometimes you'll notice that when you get these, there are very tiny, hard balls like this. But if you soak them in water, they can actually turn into this, which are kind of like these jelly, almost like boba balls. And that's because they are also made with the same material. They are made with sodium polyacrylate. So they are capable of swelling with water just the way the gel does. And so as a result, leaving them in water for a while, you'll get these squishy balls like this. So now that we've taken a look at our hydrogels, I'll just send it back over to Liv, and she can tell you some more. So thanks, guys.
I think I'm going to present next year.
Oh,
so, okay, so hi everyone. My name's Ty Karaba. I'm also in the chemistry department here at UCLA. And today I'm gonna so far we've kind of learned a little bit about polymers. We've learned about hydrogels, the kind of interactions that polymers have with water. But now we're going to talk about how we can actually control the amount of that hydrogelling, or how we can take a liquid polymer like in these linear chains and go to an actual gel solid, and how we can control the extent of that. So to do that, we're going to do a demo today showing the spherification of juice, or making popping boba. These are kind of like caviar, if you guys have ever had those, or the popping boba that you see at frozen yogurt places or things like that. So before I actually get into the demo, I just wanted to give a brief background on some of the science of how this works, how we can do this controlling of hydrogel structure, not just have the hydrogel already made like Diane showed, but actually controlling the extent of that gelling. Just to briefly go over what we're going to actually do in the demo. It's very simple. We're going to start off with some sodium alginate, which is similar polymer to what Diane showed here. You can buy it food grade. I bought this off Amazon. You'll see it's a similar structure to start with, where we have, it's just kind of a fine powder. So we're going to take that sodium alginate, mix it with distilled water and some juice, Kool-Aid, or I have just some fruit punch powder that you get at the grocery store. So that's kind of one element of this reaction that we're going to be doing. And then we're going to drop little droplets of that into a calcium chloride bath. So calcium chloride is a chemical very similar to sodium chloride, which you guys see all the time. You eat it all the time. That's just table salt. Calcium chloride is also a type of salt. And so what we're going to do is drip this solution that we have right here, the sodium alginate and juice, into a calcium chloride solution, and we're going to get out these nice spheres, and they're going to be solid spheres that we can kind of control how much liquid's inside them. And so the question is kind of how this exactly works and how we can control that amount. So just to go into what the chemicals are that we're going to be using here, sodium alginate is a biopolymer, actually. So this is the first biopolymer that we're using. It's found in brown seaweed, kind of what gives seaweed and kelp the the structure that it has, but still maintains its flexibility so it can wave in the ocean currents. And the structure of this polymer is a little bit complicated, but pretty simple. Like Liv was saying before, a lot of polymers that you see in nature are made up of these sugar monomer units. So that's this kind of complicated molecule. Don't worry too much about that. It's made of this backbone of sugar that are all negatively charged. So that's important for what we're doing next. And the sodium element is just balancing out that negative charge with a positively charged sodium. And so along this molecule, for every one monomer unit you also have one little sodium ion that's balancing that charge out. So that's how this this sodium alginate looks. And so, okay, so to go into the actual chemistry, the reaction is very simple. We're going to start out with this sodium alginate, mix it with calcium chloride. And like I said before, sodium and calcium are pretty similar. They both make salts with chloride. All that's going to happen here is you switch out the sodium and the calcium. So now the calcium will form with the alginate and the sodium will just make table salt. But the interesting thing here is that we're going from sodium alginate, which is a liquid. I have it right here. You can see it's a liquid. It's a slightly viscous liquid, but it's definitely liquid, to something that's much more solid, these polymer spheres. There we go. Okay, so just to go back to the basics of chemistry, let's look at the periodic table here and look at where sodium and calcium are in relation to each other. So if you guys have taken a freshman or basic chemistry class, you should know that this first column on the periodic table here, these are called the alkali metals. And the second column is the alkaline earth metals. Those two names are pretty similar because they do kind of make similar compounds. You saw both of these make salt, a type of salt. The thing I didn't mention is calcium chloride also you can buy at Food Safe. It does taste salty. It tastes a little bit less salty, but you can still eat it. But the difference here is that calcium is in the second group, sodium is in the first group. And so sodium will tend to make ions of a +1 charge. So it makes sodium +1 when you put it in, when you dissolve it in water. Whereas calcium, since it's in this second row, it has an extra electron to give away. It tends to make this 2+ charge. And why is that important? Well, if we go back to the equation from before, if you guys have taken enough chemistry to notice this, you'll see that that equation that I showed before was not balanced. It wasn't a balanced equation. But here now, if we actually balance it, you'll see for every one sodium alginate monomer, or for every one calcium that we put in the solution, we actually need 2 sodium alginate monomers, because this is just that +1 charge, whereas calcium is +2 charge. So sodium bonds to 1 monomer, calcium will bond to 2 monomers. And that's the really important thing here, and to how this gelling forms, is that the sodiums will get swapped out for the calciums because they're +2 charge. They want to bond a little bit stronger, and those calciums can now bond to one polymer over here and another monomer over here, and thus linking those two polymers together. That's what we call cross-linking. And so if you get enough of that cross-linking to happen, you actually form a gel structure. And so, yeah. Okay, before I go into the next one, let's go ahead and actually do the demo here. So I'm gonna tilt this down so you guys can see here. So I already have here a sodium alginate mixture that I mixed up with some of the fruit punch powder that I have. One thing I didn't mention is you do need to blend this stuff in a blender. It doesn't need to be quite as hefty as this, but the sodium alginate takes a little bit of time to dissolve because it's so thick. So here I have this sodium alginate juice solution, and here I just have a calcium chloride in water solution. And so now all you have to do, I put it in this little kind of hot sauce sort of dispenser bottle. If you have like a syringe or just a dropper or anything like that, that'll help. Or you could actually even just drop it off a spoon. But to make the best spheres, you really want kind of something that can let nice even sized drops out. And then all you do is just drip this drip the solution into the water. I don't know how well you guys can see this, but if you leave it in there for, I'm going to leave these first ones in for about 30 seconds or so. And while you're doing this, you can play around with kind of the height that you're dropping it from. If you drop it from really high, they tend to make these like flat, more discs. And if you drop it low, they kind of sometimes have little tails. So I'm going to take those out and just rinse them off in a little bit of regular water with no calcium chloride on the inside. And then now you can see we have made these little nice polymer spheres. And so I'm going to go ahead and show you. While it is a solid ball, you know, I can run this around through my fingers. If I try to pop it, it actually squirts out some juice. I don't know if you guys could see that here. So it is a popping little boba. But here now I have one that I made at the start of when Diane was talking. I left that in the solution for 5 minutes instead of 30 seconds. And you can see if I try to pop this one, nothing comes out. All that happens is you get this, you can break the polymer, but it just kind of stays in this jelly, goopy state. So that's something you guys can play around with different times too. So, you know, I only did 30 seconds and 5 minutes, but depending on the different times that you use, let me go back to this, this presentation really quick. So when you're at your home, when you're doing this by yourself, if you want to try it out, I would suggest doing it for kind of different amounts of time, 30 seconds, a minute, 2 minutes, and 5 minutes. And then one thing that you can do to, you know, instead of just kind of testing to see how much liquid is by popping it with your finger, one thing you can do is try and stack coins like quarters or pennies or something on top of these balls. And you should be able to see that for your 30 seconds, it should pop with, you know, just a couple pennies. And as you go longer and longer, as you leave it longer and longer in solution, it should be more and more solid. It should take more and more coins to pop. And so that's a good way of like kind of more quantifying how the difference in gelling of what's happening here. And so the reason why this is going on is this calcium, as you drop the spheres into the solution, the calcium has to diffuse into the inside of the droplet. And so at first, it's just reacting with this outer part of the droplet. So that's what makes that gel shell around the liquid on the inside. But as you leave it longer and longer, the calcium diffuses farther and farther in until you get a fully solid sphere at the end, like this one that I showed you before. So there's no liquid left on the inside after long enough time. Okay, so this is just showing one way that we can, you know, as humans, control the extent of this gelation in the biopolymer. But you can think about this in biological systems as well. You know, they can take up different amounts of calcium and cause this different amounts of gelling in the alginate itself.
So the calcium.
Okay, so sorry. If you have a question, please write it in the chat, and we'll try to answer the questions in the chat. And so lastly, I just wanted to show you guys, if you want to take a brief second, we have a nice YouTube video that kind of shows what's actually what's actually happening, the atoms moving around and reacting here. So this is just a little QR code if you want, and I'll post the link to this video too when we send out the instructions for later. Okay, so that's just showing that you know, how we can control polymerization with a biopolymer. But another thing that's also used a lot nowadays is, you know, actually controlling gelling in a non-biological polymer. And so in that, Liev is going to go ahead and show you guys a cool demo on how to do that. One way that we can do that as scientists too.
Hi again, everyone. I'm going to take a second just to make sure that you guys can see both my video and the screen that I'm sharing, because I do have a PowerPoint that helps. All right, so you guys have seen an example of a polymer interacting with water and ions in the case of Diane's diaper example with sodium polyacrylate. And you guys have also seen how cross-linking can occur in a biological polymer, such as with the sodium alginate. But if you think back to the slide I had with all the different examples of consumer products, you'll see that polymer cross-linking can lead to a variety of different physical properties. So if you guys have ever played with slime, you know that it's kind of a weird one. Sometimes it seems like a solid, sometimes it seems like a liquid, depending on the type of stress you're applying to it. This is what we call a non-Newtonian fluid. And I'm going to show you a really easy way that we can prepare slime with some materials that you guys might have lying around your house. So the 3 ingredients I'm going to need are glue, baking soda or baking powder, which is what I'm going to be using, and contact lens solution. So to start with, we have glue. I'm using Elmer's glue, which is PVA based. That stands for polyvinyl acetate. As you know, poly indicates that it is actually a polymer in itself. And I'm going to pour out 1 tablespoon. So this glue that I'm using, it's a polyvinyl acetate base, but it also has some other ingredients. It also does have a component of water, which as you are trying to glue things together, you usually have to wait for that to evaporate to leave your strands of polymer. All right, great. So I have my glue in there. And then I'm going to go ahead and add half of a teaspoon of baking powder. And I'm going to add it directly in there. All right. So what you're going to want to do is mix your baking powder and glue thoroughly. The purpose of the baking powder, or baking soda if you're using that, in which case you'll want to scale it accordingly, is that it actually contains sodium bicarbonate. So the sodium bicarbonate is going to be raising the pH of my PVA glue. And what this does is it's actually converting some of the groups on the acetate chain into polyvinyl alcohol, which is going to allow them to be cross-linked more easily by our 3rd and final ingredient. So I'm just making sure that I have this thoroughly stirred. I will add a food coloring. Right now it looks a little bit like whipped cream. So if you do have food coloring, right now is the time that you would be adding that, and whatever you want to, whatever else you have. If you have any craft sparkles, that's something else you can do. This glue's kind of sparkly already. Okay. So now that I have that thoroughly mixed, for my last ingredient, I have contact solution. And you don't need a lot of this, thanks to the sodium bicarbonate and the baking soda. I'm only going to add about 1/3 of a teaspoon. All right, so pour that right in there. And then you just mix it. Baking powder, the difference between that and baking soda, the baking powder also contains starch, which is a polymer itself, a biopolymer actually, that can also be used to thicken water or any stews. So here's some starch. So adding baking powder as opposed to baking soda should only make it a little bit gunkier. All right, so as soon as it gets pretty hard to stir, that's when you can start thinking about just getting in there. All right, so now you have your slime. So what's really going on here is that we have the polymers, the long chain polymers, the polyvinyl acetate that's in our glue as it is. And then the sodium borate in the contact lens solution is actually creating these polymer networks. And in this way, we go from something that's a little more liquidy to something that has, I guess, more solid. Pretend that this is slime and not really sticky. So with this, you can imagine that with a variety of different materials, as well as mixing ratios and procedures, people are able to make all sorts of materials with the desired mechanical properties. And you can think of a couple things that you might find useful with a solution like this that is sometimes behaving like a solid, sometimes behaving like a liquid. And of course, if you were to add more cross-linking reagent, you would get something that could be much harder, even like a plastic ball in that case. And pretty much immediately, I can feel that it's thickening up as this cross-linking is occurring. So what my recommendation is, if you're using this recipe, a lot of other recipes that you might find online for slime use something called borax, which from the name you can guess is also a source of these borate ions. I found that that was personally harder to find in stores as opposed to contact lens solution. So this is just an alternate recipe, but the chemistry is the same. And what you're gonna wanna do at this point is just stir it up until you get the consistency that you're looking for. So I usually mix it evenly, and then I'll add another tablespoon. All right, so now I'm really feeling it start to thicken up. To save you guys the boredom of just watching me stir this, pretty much you just repeat this process until it thickens up. At a certain point, if you're mixing it in a cup, you'll find that it almost becomes impossible to stir with a stick. And at that point, you can just reach in there and grab your slime out and then start kneading it by hand. I made a little bit right before this demo actually, and I put it in this Tupperware, and you can see it almost looks like a liquid. It's taken the shape of the Tupperware. So make my video a little bigger. It's taken the shape of the Tupperware, but then if you poke at it, it's like a solid, and you can just pick it up. So what slime is, now that you've seen the chemistry, is a non-Newtonian fluid. And what this means is that the viscosity of it is not linear, and it depends on the amount of stress that you're applying to it. So another way of thinking about that is that sometimes it seems like a solid and has a shape, but other times it has properties like a liquid and just flows. So you can think about why this might be now that we've looked at properties of a hydrogel. We have these cross-linked polymers, but we also have about 90% water involved in this glue. So if you do leave your slime for a while, it will dry out and get like a harder texture. And that's just because the water helps these polymer strands slide around and gives it this liquidy texture. So it's pretty cool to play around with. But you might be thinking, slime is pretty cool, but you know, why would you want to make this? What sort of advantages does this have scientifically or for real world applications? So kind of a way of framing what I mentioned earlier, I'm going to share screen again to show you guys my PowerPoint. When we talked about how easy they are to process, one thing you can think about is how being able to tune the properties so finely can be really advantageous. Sometimes you might want something that's more flexible, like a fluid. For instance, like paint. When it's wet, you want it to be in that form so that you can paint it on whatever shape you're painting. Whereas if you have something that is, you know, more rigid, maybe this is going to be more durable. But then, for instance, if you have something like an earthquake, a bridge that's more flexible is going to be more mobile in that situation and might be more advantageous in that case. So in this kind of analogy, the pieces of wood and the planks are sort of our polymers, and then these crosses and how they're connected is the idea of cross-linking them. So now to give you guys a more real-world example of what I'm talking about, we got a question that was submitted ahead of time asking us what polymer research is like and what sort of things people do with biopolymers. So there's two examples that are actually being worked on by groups in CNSI, the Nanosystems Institute, and they have to do with both tissue scaffolds and bioinks. So in the first case, I'll give you this idea. If you're doing a surgery and you have a really small incision in something like an artery, how are you going to close that up to stop the bleeding? So traditionally, people do use a lot of physical methods, even in external cuts like stitches or something. But more and more people are starting to look into hydrogels and different cross-linkers to be able to heal these wounds and stop the bleeding. So an example might be that they inject something that's liquidy at first, but then either if it's opened up as in a surgery, they can apply something like a UV light to cross-link these polymers and help close up the wound. As well as maybe they inject a second reagent, such as like we did with the sodium borate, to be the cross-linking agent. So challenges in this case are making something that has the right durability, but it also has to be biocompatible. So you wouldn't want something that caused, for instance, an allergic reaction if it's going to be going into your body. Other examples, as you can see on the figure on the right, would be something like cartilage repair. As people get older and joints wear down, a lot of people have maybe like hip replacements, but people are increasingly looking into maybe like softer ways of achieving this healing through biopolymers that help restore those tissues, but we have to design them in a way that they're sturdy enough, flexible enough, and biocompatible. So that kind of leads me to the second example that's also being worked on by a group in CNSI. And I'm gonna have this video playing at the right that you guys can watch. And this is bioinks. So one thing, as people study more and more complex relationships of cells and organelles in the body, the traditional way of growing cell types would be taking a plastic petri dish, having some nutrients, and growing your cells in that dish. But we know that this doesn't fully reflect the complexity of the environment that cells are actually in a living organism. So in the body, you have tissue. It's like a 3D structure. And then there's other things besides the cells. There's proteins, there's molecules, nutrients, all sorts of different environments. And so to sort of create a better platform to study and mimic these things, people have started 3D printing scaffolds in 3 dimensions so that they can grow different cell types and organelles. The term organ on a chip or lab on a chip a lot of times refers to this kind of practice of making like a miniature model of a system that still has the complexity that really mimics the real system. So you can see this 3D printer is printing a variety of hydrogels, kind of showing off a little bit to show like the different materials it can make and the different shapes. You can see it's kind of turning out to be a heart. And then in these hydrogel bioinks that have that thickness, people then can grow cells to try to create these systems that are, like I said, more close to the actual environment, so that when we study things like how drugs interact with a part of the body, it's a little bit more informative than just cell in an artificial environment. And yeah, so this area of research is really cool. If you guys are interested, we can also supply you with links to see either more videos like this or examples of what biohydrogels are being used for. But yeah, so that's sort of the end of my presentation. I see that we have a lot of questions in the comments. So thank you guys so much for listening. Hopefully you're able to follow along with these demos that we showed you guys, and we'd be happy to answer some questions now.
Yes, so there were quite a lot of questions about are these reactions endothermic or exothermic? That was a very repeated question. So did you guys want to answer those ones?
Yeah, all of the reactions that we're going to be doing here are going to be exothermic, just because we're not actually supplying any energy into the system. It does the reaction by itself. So it's a spontaneous reaction, which is something you guys might have heard before. And so that's typically seen in exothermic reactions. If we had to heat this up to cause the gelation, like if you've ever made Jell-O, you actually have to heat it up first and then cool it back down to cause the reaction. That's more like an endothermic reaction.
Yeah, that's a really good point. And another way that I usually think about that is anytime you're breaking bonds, it requires mostly, unless there's some sort of pre-existing structure and a big difference in terms of the chemistry of what you're starting with versus what you're making. Generally, a good rule of thumb is that breaking bonds takes energy and you're putting energy into it. And in that case, it would be endothermic. But in this case, with the cross-linking that we're doing, it occurs because those formations are more stable. They're favorable, like in the case of the borate ions doing the hydrogen bonding. So making that more favorable bonding is going to put the system at a lower energy because it's more favorable. So that would be exothermic because you're releasing that energy as it's relaxing, as opposed to having to put energy in to force it to make the bonds. That's a really good question though. And I think that there's some products like gel packs that heat up that rely, there's probably a little more detailed chemistry going on, but there's certainly gel products that people use to heat up, and there's also like gel cool packs, but that would be more specific with the chemicals that are involved in those. But yeah, really good question.
One other question that somebody had that was an interesting one was for super absorbent polymers versus hydrogels. So are they the same, or what's the difference? And just to clarify that, so they're generally used somewhat synonymously, but the specific difference is that hydrogels become hydrogels when they're actually mixed in with the water, and when they don't have any water in them yet, they're not called hydrogels, and they're simply referred to as super absorbent polymers. But again, I think you hear these terms used pretty synonymously, so they're essentially the same thing. But technically speaking, I believe that the difference is that with a hydrogel, it's not a hydrogel if you don't actually have any of the water in it.
I see a question too here that says, how does cross-linking increase the strength of the polymers? Which is, I think, good to describe for all of these. One kind of other way that you can think about this is like, you can think about the polymer to start with. The uncross-linked polymers is just kind of a bunch of strings that are separated. And so if you have just a bunch of strings, they don't have any kind of mechanical property. I could take one string out, it's easy. But when you cross-link them, that's kind of like tying all the strings together into a sort of net or something like that. And so that's what kind of gives it its structure. And then the water actually filling in all the gaps is what kind of makes it this solid structure. So it's a combination of the cross-linking itself and the water going into the spaces in that cross-linked structure that gives it this kind of solid, bead-like form, rather than just a liquid like this, like the sodium alginate by itself. These are just polymers floating around in solution, not really interacting as much with each other.
Here's another interesting question that somebody asked. Somebody asked, is a cell a hydrogel? Which I guess is almost a kind of an interesting thought experiment in a way.
Yeah.
I think a cell has too many complex components to simply be considered a hydrogel, because it is not just made up of one polymer. So it's something that's too complex to talk about it that way. I think a better thing that you could probably classify as a hydrogel would be something like a tissue, which is what the cells sit on and live in. But I don't think the cells would be considered a hydrogel.
Yeah, I would say, yeah.
Oh, do you want to?
I would just say there's some components in the cell that might be hydrogel, but yeah, as a whole, it's made up of a lot of.
I was going to describe it as, so you have different pieces that sit within that cell, and you can think of the cellular matrix as a hydrogel. That's almost like the thing that suspends everything, but not the entire cell. It's just kind of the cushioning material inside.
Here's polymers in the cell though. Like you could think of, if you've seen diagrams of what a cellular membrane looks like, it's a repeated unit of these lipid structures. And it's generally a bicell. So you have one end that is hydrophilic on the outside of the cell, as well as in the middle, you have this hydrophobic tail. And then you have the same structure on the other side, which creates this envelope. So that's certainly a polymer in a sense, that you have this repeating lipid unit. But there's other things that make up the lipid membrane as well. Like there's proteins that someone mentioned that are across the membrane. So I wouldn't I agree. I wouldn't say that it's a hydrogel through and through, but different layers and pieces have different polymer qualities. That's kind of a very small scale, like micro network of these materials kind of coming together to work as the cell.
So another question, so one, I guess, clarifying question, is a hydrogel a type of synthetic polymer? So there's a difference between the two. So a hydrogel is a polymer that can hold a lot of water, and a synthetic polymer is a polymer that is not found in nature. So they're just two very different things. So hydrogels can be synthetic or biopolymers, and synthetic polymers can either be hydrogels or not hydrogels. So they're just two different things. Somebody else asked, are biopolymers renewable? So do one of you guys want to speak to renewability?
Are they renewable?
Yes, are they renewable?
That's a good question. I would say at that point, you're kind of looking at the sourcing of them and how you're generating them in, for instance, an industrial quantity. So if you have good farming practices, like for instance, the sodium alginate is found in seaweed. Something like this can certainly be considered renewable if you're harvesting your seaweed in a smart, eco-friendly way. Some people who have diet restrictions, like if you're vegan, for instance, do not eat gelatin because a lot of the sources of gelatin are from animals. So it depends on what the biopolymer is and where we're getting it from. You can make, there's other things that people do, not quite the same as like a hydrogel, but for instance, alternative fuel sources involving corn. That's another thing where some people say that it's very renewable, but once again, it would depend on how you're growing it and harvesting it.
Yeah,
I also see a question that I want to try to answer right away that has to do with what hydrophilic and hydrophobic is. So hydrophilic means that it is a molecule that likes to interact with water. We use the word like to kind of like personify it, but it's something that is going to interact with water, whereas something that's hydrophobic, you can kind of think in the word phobic, it's a phobia, it's afraid of it, it's a molecule that typically does not want to interact with water. And this comes down to the chemical structure of a molecule. And a really good example that you've probably seen with hydrophobic versus hydrophilic is if you ever put a drop of oil in water, you'll notice that they bead up and tend to kind of collect together and don't want to spread or dissolve throughout the water. They want to stick together. So something like that is hydrophobic. It's not wanting to disperse throughout the water. It's wanting to form these sort of shapes and spheres together so that the molecules don't have to be interacting with water as much as they can in the space that you put them in. So does anyone want to add to that, or hopefully that clears it up a little bit more with the difference between it and how you can recognize it by looking?
Yeah, I just wanted to go back to the renewable question for a second, just to answer that a little bit more clearly. Well, so most synthetic polymers that we've seen in the past and even now up to this day are made from petroleum, which is definitely not renewable. It takes millions of years to make more, and so biopolymers themselves are definitely more renewable on shorter timescales. It takes a little bit less time to grow a tree or kelp or something like that. But also there's a lot more research nowadays going into taking polymers that we can get from plants and kind of creating newer synthetic polymers that are still coming from these better sources rather than petroleum or oil.
So another another question that we have. There's quite a few. This is a great conversation, guys. So another question that we have, so one person asked, does polyethylene oxide count as a sort of polymer? Other instructors, please correct me if I'm wrong. I believe that a polyethylene oxide is just a low weight polyethylene glycol.
Yeah, they're just two words for the same thing. Yeah.
So they're basically, so yes, it is a polymer.
Usually
you
good.
Have it in the name too. Like the poly is always a good giveaway. That poly of ethylene. If you were to look up, ethylene itself is a molecule, and then the polyethylene just implies it's similar to a repeated motif of those, or theme or structure of those individual units. So definitely a polymer. Most things that have the word poly before them as the prefix are going to be polymers.
And then a few people have been asking questions about, can we replace synthetic polymers with biopolymers? And I think the answer to that is maybe and hopefully, right? So I think that's a lot of the goals with biopolymers is, can we use them to replace these synthetic polymers that are derived from petroleum and not as sustainable as biopolymers that can be manufactured from renewable source materials. So there are a lot of objects that are already replaced with biopolymers, especially in a lot of consumer products, because that's a really popular application right now. So, for things like cutlery and disposable utensils, in that area for sure, biopolymers have been pretty decent replacements. They're still a little bit expensive to manufacture and don't always perform with the same quality of properties as their synthetic counterparts. So that's something that researchers are still working very hard to do.
The one thing I wanted to add in is that if we look at the way nature makes polymers, it's spent millions and billions of years of perfecting those polymers. So the whole point of research is trying to understand how nature does these things and have it repeated in a lab. So a lot of the inspiration comes from biopolymers to get them that way.
Yeah, so it's about 3 o'clock. So I think that we unfortunately have to bring this session to the end. Before you guys all leave though, if you would not mind, please just take a minute to fill out this post-demo survey for us that we just put up for you. We really love and appreciate your feedback because we hope to do more of these, and we definitely want to make them the best that we possibly can. So before you guys head out, please fill those out. Thank you guys so much for tuning in. We really appreciate getting to have a live audience for these things. It's really fun for us. Be on the lookout for the video that we'll be sending to you once we get the recording, and also the instructions, so that if you guys want to do these same labs at home, that you'll be able to do so. And hopefully we'll see you for another one soon. Thanks, everyone.